Hi, my name is Quinn O'Connor. I'm a senior at Sarasota High School, and for my senior project, I will be comparing somatic mutations to the Saccharomyces cerevisiae genome when environmental carcinogens are applied. So the purpose of my experiment is to examine the effects of common carcinogens on the genome as cancer rates increase using yeast as my cell. This leads me to my research question, which is what effect do environmental carcinogens have on DNA structure and sequences? And if they have any impact, what carcinogens have the greatest impact? My hypothesis is that when the yeast cells are exposed to ultraviolet rays, they will have the most somatic mutations in their DNA sequence. However, I believe that all of the yeast colonies and cells that are exposed to any carcinogen will have some resulting somatic mutations too. So some background that is relevant to my project, I'm using Saccharomyces cerevisiae instead of animal cells. It is a very good viable alternative because they are both eukaryotic and have similar nucleus. And the genes responsible for DNA repair are very similar in yeast as they are to a human. They have 23% of their genes in common, and 20% of human genes that are known to have a role in disease are actually present in yeast. They also have similar meiotic and mitotic processes, as well as similar signaling protein pathways. Carcinogen exposure and cancer rates have increased significantly, which is what makes this project so relevant. And there has been a 49% increase in cancer since 2015. Look at the bottom of the slide, the group 1, impactful and common carcinogens usually are things that are in our everyday lives, such as from your car exhaust to consuming alcoholic beverages, solar radiation, tobacco, processed meat. So this is a brief overview of my methods. I will begin by culturing the yeast cells in the colonies. I will have 3 that will act as a control, and they will just stay in incubation and complete their life cycle as usual. One of them will be extracted for electrophoresis, and I will use a transilluminator to view the initial DNA patterns so that I can view any modifications after carcinogen application that may occur in their migration patterns. I will be using UV light thermal radiation and mimicking alcohol usage and car exhaust. So this is the timeline for my project. I expect to be done by January 23rd. I begin on January 11th, assuming that all of my materials or enough materials that I can begin to get started would have been delivered. I will begin culturing and incubating the colonies at this point, and I will run a test run with the electrophoresis chamber, which I'm already familiar with, but just to ensure that everything is still working so that does not cause an issue when I am ready to collect more data. I will then calculate the cell densities on each of the dishes after there has been an incubation period of about 24 to 48 hours, which I will monitor in class. I will run one strand through electrophoresis, as I mentioned previously, to set a base for collection in the future. And then I will make my 2 solutions if time allows this day, so that I can use them in the future for my carcinogen application. Or I can do them on the next day as shown on step 3 on January 14th. I will begin the UV light and thermal radiation trials. These are relatively hands-off, so this is another time for me to make my solution. I will have them incubating for 2 days after the carcinogen application, but they will sit in direct exposure for 1 hour. I will then, on the 18th, recalculate my cell densities from that trial. I will conduct electrophoresis on the UV light and the thermal radiation trials, and I will log my data. I wanted to do this after incubating to give the cells time to regroup and to have the most accurate measurement of my cell densities. As you can see from step 5, on January 19th, I will do my chemical trials and run electrophoresis. They will be incubating for 3 hours. So I would begin around 10 a.m. and be done by around 1, depending on how time allows. And then my data collection would begin on January 20th and continue over that weekend. And I would compare all of my electrophoresis results, compare the control colony with what I had found, and also compare all of those cell densities. As I've already touched on, this is some of the ways that I will be collecting data and evaluating my results. I will be looking at the cell density of the colonies, first ensuring that there are not too many cells on one petri dish that could interfere with my results, but also making sure that at the beginning they're all relatively similar and viewing whether or not I have abnormal regeneration or if I have a higher mortality rate in those cells. I will also be looking at their macromolecule patterns and ensuring that there is a change, which would indicate that the carcinogen is altering the genomic sequences. And I will be repeating trials. This is a mock graph that I've created with my predictions on how I think each of the carcinogens will affect the average cell density. I used the control as 1.126, and I have compared this to a few different studies that can also be found in my bibliography to determine where I think there will be the most effect and where I think there will be the least effect. Thank you. That is my project. I look forward to beginning, and I will have all of my data in by January 23rd. I am looking forward to getting started.